students, this module is for Bachelor of Science, second year, subject Botany, semester 4, paper Plant Physiology, BOC 104. The title of the unit is Plant-Water Relations, and this is the second part of module Transpiration, module number 6. I am Miss Marina Janice Rodriguez from DCT's Dempi College of Arts and Science, Miramar, Goa. In this module, we are going to see the introduction, movement of water from roots to the stomata, mechanisms of opening and closing of stomata, theories explaining the mechanism of transpiration. At the end of this module, you will be able to describe the movement of water from the roots to the stomata of the plants, explain the mechanisms of opening and closing of stomata, also explain the theories of mechanism of transpiration. In the previous module, you have learnt about stomata and the function carried out by it, that is transpiration. Transpiration is the loss of excess water from the aerial parts of the plants in the form of water vapor. In this module, we will go through a journey of the movement of water right from the roots to the aerial parts of the plant and finally out in the environment in the form of water vapor. Water enters the plant root through the root hair zone. The root hairs increase the surface area for absorption. Once inside the root, the water makes its way through the plant cells in two ways, either in the apoplast pathway or the symplast pathway. The apoplast pathway includes wherein the water moves exclusively through the cell wall without crossing any membrane, whereas the symplast pathway, the water travels from one cell to another via the plasmodesmata. It finally makes its way to the xylem tissue of the plant. The xylem tissue consists of two important elements, that is tracheids and vessel elements. In most of the plants, the xylem constitutes the longest way of water transport with great efficiency. The pressure gradient needed to move water through the xylem to the aerial parts of the plant results from the generation of negative pressure at the top of the plant. The tension needed to pull water through the xylem is a result of evaporation of water from the leaves. In intact plants, water is brought to the leaves via xylem of the leaf vascular bundle, which then branches into fine, intricate network of veins through the leaves. The water vapor then exits the leaf through the stomatal pore. Mechanism of opening and closing of stomata. As you can see in the picture, it shows an outer structure of stomata. The adjoining pictures, that is in black and white, show two different stages of stomata, that is the opened stomatal pore and the closed stomatal pore. When the guard cells of the stomata become turgid, the stomatal pore opens. When the guard cells are flaccid, the stomatal pore closes. The outer wall of the guard cell is elastic, whereas the inner wall is thick and non-elastic. When water moves inside the guard cell, it becomes turgid. The outer wall expands. Along with the outer wall, the inner wall too expands, and hence it leads to the opening of the stomatal pore. When the water leaves the guard cells become flaccid, and the outer wall as well as the inner wall regains its original shape, thus closing the stomatal pore. Many plant physiologists have given various theories explaining transpiration. The first one, theory of photosynthesis in guard cells. Von Mohl in 1856 observed that stomata open in light and close in the night. He proposed that photosynthesis in the guard cells, the chloroplast present in the guard cells, carry out photosynthesis in the presence of light. This leads to the formation of carbohydrates, which increases the osmotic pressure of the cell sap. Thus, It leads to the end osmosis, wherein the water moves inside the guard cells, thus making it turgid. It increases the turgor pressure in the guard cells to open the stomatal pore. The next theory is starch-sugar interconversion theory. The insoluble starch present in the guard cells is hydrolyzed into soluble glucose 1-phosphate in the presence of phosphorylase enzyme during the daytime, and soluble glucose 1-phosphate is converted back into insoluble starch during the night. Stomata open in neutral or alkaline pH, which prevails during the daytime due to constant removal of carbon dioxide by photosynthesis. Stomata remain closed during the night when there is no photosynthesis, and due to the accumulation of carbon dioxide, carbonic acid is formed that causes the pH to be acidic. The third theory is potassium pump theory. During the daytime, starch is converted into malic acid in the guard cells of the stomata. This decreases the starch contents of the guard cells in the presence of light. It also leads to the increased concentration of malic acid in the guard cells. With the production of hydrogen ions, there is influx of potassium ions in the guard cells in exchange of the hydrogen ions thus formed. With the influx of the potassium ion, there is also intake of chloride ion by the guard cells. This leads to high pH in the guard cells, which increases the turgor pressure in the guard cells. Thus, there is movement of water inside the guard cells, making the guard cells turgid. It leads to the opening of the stomatal pore. Let us see what happens during the night time. During the night time, there is decreased concentration of malic acid in the guard cells. Thus, there is less concentration of hydrogen ion. This leads to the efflux of potassium ions from the guard cells and influx of the hydrogen ions in the guard cells. It leads to the acidic pH of the guard cells. Along with the efflux of potassium ions, there is loss of chloride ions from the guard cells. It increases the carbon dioxide concentration in around the guard cells. This is because there is no photosynthesis which occurs, and also there is respiration which is taking place. In the presence of plant growth inhibiting hormone, that is abscisic acid, there is loss of turgidity and thus leading to the loss of kidney-shaped guard cells. It leads to the closure of the stomatal pore. These are my references. Thank you.